Welcome to Ajad Bioacademy. Today's lecture topic is agonist and antagonist. Okay, so today I will discuss agonist and antagonist. In my first lecture, I will cover agonist and antagonist. Then I will explain the types of agonist in second lecture, and the third lecture types of antagonist, different types of antagonist. Okay, so agonist and antagonist. Before going to details, I will just give you some basic idea which are very much related to agonist and antagonist before going to the main discussion. So first one is receptor, and second one is ligand. All right. So receptor means the macromolecules, basically glycoprotein in nature, but also you can say protein in nature, which are mostly present as a transmembrane form, sometimes also present inside the cell or also outside the cell. So why it is called receptor? Because after binding ligand or some substances, the receptor has ability to change its conformation, and then it will give biological response. That means on which drug or ligand will bind and give its response. That is called receptor. And ligand is any endogenous or exogenous substances which will bind with the receptor and which has ability to change the biological response, change the conformation of the receptor, or which can block the receptor. That means which will bind the receptor, that is the ligand. Okay, so just I will give you a brief discussion on receptor and ligand through a figure. Okay, so here you can consider as a membrane, and this you can consider as a receptor. So this is receptor. This transmembrane form is a binding site, the extracellular part, and this is the signal sequence, that means stimulatory site that is inside. Okay, so if it is considered as a receptor, then I say something will bind here. You can consider as a binding pocket. So here it will come here, and it will form a bond with that, and it will change the conformation of this receptor. And with the receptor, had some enzyme or ion channel or some proteins are attached that will be activated, and it will give the biological response. So this protein is called receptor, and that substance is called ligand. That means ligand are the substances which will bind with the receptor, and receptor are the substances which will receive the drug, and through the reception it will give its biological response. Okay, so this is the matter of receptor and ligand. Why I am just discussing other than agonist and antagonist, then I will come here now. So now come to the point, agonist. Agonist means what? If I just try to explain, that is, agonist means it has attraction to the receptor and it has ability to change the conformation of the receptor. That is called agonist. So agonist can be endogenous or exogenous. You can say ligand. Okay, so some ligands, endogenous ligand, I can give you some example like hormone. You can think about hormone. You can think neurotransmitter. You can think autopoiets, or the endogenous ligands that will specifically bind with a specific receptor, and that will change the conformation of the receptor and gives it their function. Okay, so suppose if you think epinephrine or adrenaline just increase the heart rate, that will bind with the receptor, beta 1. If you think some neurotransmitter like acetylcholine, dopamine, they will bind the corresponding receptor and give their function. If you think about what occurs, that is serotonin, or you can say histamine, that is also a specific receptor, and they will give its function. So these are the ligand. Okay. So now the ligand will be agonist, and some similar substances which will be applied from the external source, that means as a drug, that can be each other as an agonist, or some endogenous substance. That means two substances, their binding site and their activity is similar. So that will be the agonist to each other. So now I will come to the point here, that is one parameter, one criteria must have, that is criterion, that would be the affinity, and the second one would be efficacy. Okay, so what does it mean actually? Affinity means the attraction, binding ability. That means the receptor has a specific binding site, that is binding pocket, you can say, and the agonist must have the similar structure to fit in that area. So the size must be like that. Okay. As a result, it will completely fit with here, fit with it. Okay. So this is the criteria. That means its shape and size must be like to bind with that area, and the molecule should be flexible to fit very perfectly with that binding site. Okay, so it has to be like similar structure, or you can say particular structure, or it must have ability to bind with the receptor. So what would be the ability? It means the agonist, the chemical substances or drugs, that must have a specific functional group. And you know, the receptor is made up of protein, that is the sequence of amino acid. Some sites are there, some amino acid must meet a criteria to form a bond between the ligand and the receptor. That can be van der Waals force, that can be a hydrogen bond, that can be covalent bond, any other bond. Okay, so the bond forming ability must be present between the drug and ligand. Okay, so what would be the affinity? Affinity means the attraction towards the receptor and ability to form a bond. That is the affinity. Okay, and another one is efficacy. What does it mean, efficacy? Efficacy means, okay, it will just come to the receptor and it will bind with the receptor. That's it. Now, after binding, it must have the ability to just change the conformation of the receptor. Okay, it will be able to change the conformation of the receptor. When the receptor conformation will change, then you know the receptor is basically attached with some protein. That can be, you can think that can be G protein, or you can think that can be enzyme, or you can think that can be attached with some ion channel. Okay, so when the conformation will change, then the attached enzyme or attached G protein or attached ion channel will be activated. Okay, so for the biological response, that type of protein or enzyme or channels must be activated. And who will activate? The activate will activation will be done by the receptor. And who will change the conformation of the receptor? That is the drug, ligand, or agonist. So agonist must have the ability to change of the receptor conformation. So what is the ability? Ability means that is the intrinsic ability. That means inner ability to change something. Okay, so if you just ask me to hold something, so I must have energy to hold the substance. Okay, that is my inner ability. But if my hand is paralyzed or neural problem, then I will not be able to hold something. Okay, so suppose my hand is like that, and if you just put something in my hand, so if I'm not able to just fold my finger to hold the substance. So then that means I have the ability to capture it, or if I am unable to do that, that means my hand is paralyzed, and when you put something, the hand will be unchanged. Okay, so efficacy means intrinsic ability or inner ability to change the substances, the receptor. Okay, so that is the efficacy. So agonist must have the ability to change the conformation. Okay, so the inner ability is efficacy, and only the binding affinity, binding attraction, or binding capability is affinity. So agonists have both criteria. It must meet the criteria to form a bond, or that means binding ability, and also it will capable to change the conformation of the receptor. And which criteria will change the conformation? Because it has a specific, very quick binding frequency, and that will easily change that will be very flexible, and that will easily find out the exact location to bind properly, and it will change the conformation. Okay, so that means agonist has a very flexible, agonist has a specific shape and site with the receptor binding pocket, and it will best fit with the receptor to change the conformation. So that criteria will help the agonist to show its efficacy. So ultimately, efficacy has the innermost ability. Okay, so a specific molecule has a specific ability. Okay, so now if I just try to give an example, that would be very better. That is, if you think like, I will just give an example now. Okay, so here you can think just a similar structure like you can say this is adrenaline, or you can say epinephrine, and the similar structure you can think this is, oh sorry, this is suppose you can think this is the beta 2 receptor, and this is the epinephrine, and this is, you can say this is salbutamol. Okay, so when adrenaline or noradrenaline will bind with the beta 2 receptor here, it will bind here, it will change the conformation, it will activate the receptor, the beta 2 receptor basically present in lungs, and this is the G protein coupled receptor. The G protein will activate and it will causes bronchodilation. Okay, because epinephrine or norepinephrine will bind properly with the beta 2 receptor, and it will change the conformation and gives the biological function. And this one is the drug that is externally you are just taking when you are suffering from asthma or difficulty of breathing. Then you will, of course, you will take salbutamol. Then salbutamol will bind the same receptor, and it has also ability to change the conformation. That means the similar activity and the similar structure. So this one is agonist with that That means both are agonist to each other. You can say also this one is agonist with the specific receptor, this one is also agonist with the specific receptor, and both are agonist to each other. Okay, so this is the agonist method. So agonist has the ability to change the conformation and has affinity toward the receptor. If you just I'm just try to explain by the lock and key theory, that would be also easier. So suppose if you think this one is a lock, okay, and if you think this one is a key, so suppose if you think this one is just I'm taking that way, that is black color, that is salvator, and this one is same structure, that is noradrenaline, or you can say adrenaline, or this is salvator. If you just try to just unlock the key lock, then both keys are able to unlock the lock. Okay, if you just try to open the lock, you will be able to open through this key, also through this key, because both keys will properly fit with the core of the whole of the keys, and both has a particular teeth that will just change the inner withstand force to open the lock. Okay, so you can say both are agonist to this lock and both are agonist to each other. Okay, if you just give another example, like suppose if you think this one is a particular chair, and in a office someone is just playing a role as a chairman. So if you think this one is a chairman, okay, this one is a chairman. So this chairman, he is capable to just perform his duty, official duty. Okay, if vice chairman is go there in absence of chairman, so vice chairman, this is, you can think this is a vice chairman. Okay, this vice chairman also able to perform the official duties, and he is very eligible to sit on the chair. Okay, because he is eligible, he has made the criteria. So this is vice chairman role. Okay, if someone peon is try to just sit on the chair, then the peon will be able to perform the official duties? No. But peon has the particular affinity. Okay. If I just get a chance to sit on the chair and day. So peon, if you just give chance, okay, go to the chair and sit and perform the duties, that peon will not be able to do that. Okay, so chairman and vice chairman both are agonists to each other. The chair is fit for all of them. Okay, so this is the agonist method. Okay, now I will try to explain the antagonist. Antagonist means, now anta, which is anti-agonist. That means function is completely opposite of agonist. So why it is antagonist? Antagonist has one thing, is basically it has also affinity, as agonist, the structural similarities. But in particular area, if you just look closely, then you will try to find out the structural differences. Okay, that means roughly the structure similar with the agonist. That means it will meet the criteria to form a bond with the receptor. That means the binding pocket, the size of the antagonist is as like as the agonist. Okay, so antagonist has affinity. Okay, as like as agonist, but antagonist has no efficacy. That means antagonist has no any capacity to change the conformation of the receptor. Just it will just encroach the proper binding pocket, and it has no any ability. Okay, so how will you explain? That means antagonist has no intrinsic ability to change the conformation of the receptor. That means you can say this is inactive, but it has affinity towards the receptor. Okay, it will meet the criteria to form a bond with the receptor, but has no best fit with the receptor to change the conformation. Okay, now if I just give some example, that would be very easier for you to understand. Okay, so now I will change the example. Suppose if you think this one is beta 1 receptor. So you know beta 1 receptor is very much rich in heart and also kidney. So if you can consider this is heart, and this one now you can consider as a atenolol. Okay, so you know this is a beta blocker, selective beta blocker. So that beta blocker will structure is as like as agonist, although innerly somewhat different. So here it will bind here like that. It will form a bond with the receptor and has no any inner ability to change the conformation of the receptor. Okay, that means it will just encroach the position. It will just sit on the binding site, but has no ability to change the conformation of the receptor. That means no efficacy. Okay, so this is the antagonist. Why it is called antagonist? It will antagonize or anti-agonist. That means when the noradrenaline or adrenaline will bind here, it will form a bond and it will connect properly with the inner best fit of the receptor, and it has ability to change the conformation of the receptor, and it will activate the connected protein or enzyme or channel, and that will give its function. So now here already I consider as beta 1 receptor. You know beta 1 receptor is G protein coupled receptor. So when noradrenaline or adrenaline bind with the beta 1 receptor, it will change the conformation of the receptor, and G protein will activate it and give its function. It will increase the heart rate. Okay, and if you just in the intake atenolol, the atenolol will bind properly with the receptor, but it will not be able to change the conformation of the receptor, so G protein will not be activated and no biological function. That means that atenolol will just antagonize the noradrenaline or adrenaline. So this one is each other antagonist. Okay, so if I just closely give example with the lock and key, so that would be very easier for you. So suppose you think this one, the black one is your atenolol, and red one is your noradrenaline. Okay, so noradrenaline we easily if you just put here, it will properly enter into the log, and it has some specific teeth that will best fit with the inner part of the receptor as like lock, and then if you just twist, then lock will open. That means that noradrenaline has ability to open the lock. But here, if you just look closely, the whole structure of the key is like as the noradrenaline key. But when you put here, it will enter, but it has no specific teeth, and it will not be able to open the lock. Okay, so it has ability to enter. That means ability to bind with the lock. That means has affinity, and it has no ability to open the lock. That means it has no efficacy. Okay, and if you think this one is the chairman, and it will be able to sit, it will perform the official work as a chairman by sitting on the chair. And if you think this one is a peon, so peon has no ability. Peon will easily can take the chair, but peon has no ability to perform the official duty. Okay, so the peon will antagonize the chairman, because when peon will just sit on the chair, then the chairman will not be able to sit at that time. Of course, the peon must leave the chair. Okay, so in that way, if we just think in another way, that means in Indian subcontinent, you know, the goat, the male goat, the scrotum is removed, that means castrated. Then it will make as like castrated goat, male goat. The castrated male goat has affinity to the female goat, but has no any ability to do this activity. Okay, but if you think the non-castrated goat, that means female goat, which is completely okay with the functioning scrotum, and it has affinity to the female goat, and it has also ability to do its activities. So that is the castrated goat is antagonist, and also you know the complete goat which has completely functioning scrotum, that is the agonist. Okay, so just I will just give you an inner way, that is agonist. Like if you think this one is like a receptor, and if you just make a design of the antagonist, that is basically something roughly something like that. That is agonist bar antagonist. But if you just try to make a design of agonist, that would be like that. Here are some points. So your agonist structure should be like that. Sorry. That means it will easily fit properly with the receptor. But here it will bind, but has no properly attached with the inner part of the receptor. That means initially I covered, that is agonist will be flexible, will be similar shape and size, and it will frequent binding frequencies, binding is high, and it will quickly attach with the receptor properly, and that will change the conformation. But antagonist will bind with the receptor. It has also binding capability, but has no best fit with the has no ability to best fit with the receptor as agonist, and it has no any intrinsic activity. So that is the antagonist. So this is all about agonist and antagonist. Thank you very much.